Hello and welcome to a brief introduction to transcriptional regulation, eukaryotic transcriptional regulation to be precise. And what we're going to start off with is we're going to start off with a brief introduction into transcription, a nice little refresher. And so basically transcription, we're going to start in the initiation phase. In the initiation phase, we're going to get our transcription factors. We're going to get them to bind upstream of where we want to, what gene we want to encode. We're going to get them to bind upstream. And then once all the transcription factors are in, we'll still be in the initiation stage where everything's set up. We're going to get our RNA polymerase, and it is going to come and bind. It's going to bind between the -10, so 10 base pairs upstream of where you want to bind. It's -10, also called the TA box, and the -35 box. So from -10 to -35, it's about 25 base pairs long RNA polymerase. And so what we're going to do is we're going to get that to bind there, and then after all of our transcription factors and everything is in place, we are going to enter the elongation stage. And the elongation stage is we're going to just use this template strand of DNA, and we're going to use RNA polymerase, pry it open, and run along and make a complementary strand. And since DNA is 3' to 5', our complementary strand, we're going to be made 5' to 3'. And so we're going to go and we're going to make, eventually we're going to hit a stop codon, and RNA polymerase is going to stop, it's going to leave, and these transcription factors are also going to leave, and then we're going to have our strand of mRNA. And our mRNA consists of two components. It consists of introns. This is information that we don't really want, we don't really need, and exons. These are the money makers. These are the ones that we actually want. And so we're going to get spliceosomes, and what they're going to do is they're going to splice out the introns because we don't want these, and they're going to splice together the exons. And so now we have a complete mRNA. And then with the mRNA, we're going to protect it with a 5' cap and a 3' poly A tail, and we are going to send it out into the nucleus, where it will go. It'll find a ribosome, and then it will start making a gene. It'll start translating, and it'll start making a protein, which will go on and do things in the body. And so, how we can regulate this? Well, eukaryotes, we have some funky stuff that we can do. So we have these things called effector molecules. And effector molecules are split up into two groups. The effector molecules, we call them activators and repressors. And so what the activators are going to is they're going to speed up the reaction. They're going to make it go a lot quicker. They're going to get RNA polymerase to come on over, and we're going to get to bind much quicker and go much, much faster. We also have things called our repressors, and much like the name, they repress the gene. They make things go much more slowly, and sometimes they completely outright stop RNA polymerase from coming over and binding. And some common types of activators and repressors, they have specific functions and specific shapes. They're called like zinc fingers and leucine zippers. Those are two of the very common ones. And what the effector molecules are, activators and repressors, what they're going to do is they're going to bind to the control elements. And what are control elements? They're called the enhancers and the silencers. And so an activator is going to bind to an enhancer, and a repressor is going to bind to a silencer. And the enhancers and the silencers, they're just areas where our effector molecules will bind to and will either speed up or slow down a reaction. Now, another way that we can use to regulate eukaryotic transcriptional regulation, another way that we can regulate it is with something called silencing RNAs. And so what silencing RNAs is a piece of RNA that's different from the one we made. We inject it from outside the cells. Viruses can do this. Scientists could also do this in a lab. And what they do is they're complementary with themselves. And so this first bit is going to be complementary with the second bit. And so eventually the RNA is just going to want to come on over and bind with itself. So it makes this nice little double strand with what we call a hairpin. And so what this does is going to bind to a specific protein, and it's going to search out specific mRNA that we've created, and it is going to go and it is going to destroy them. The mRNA is going to bind, the protein is going to destroy them. And so that's another way that we can regulate things, and is after we transcribe something, we can destroy the mRNA, so it never goes out into the cytosol, um, never finds one of the proteins to bind to, and it never goes through translation to make a protein. So that's another way that we can regulate eukaryotes transcriptionally, is through after we make the mRNA, we're going to get that silencing mRNA to bind to it and then get a protein to destroy that. And that was a brief introduction to transcriptional regulation.